we'll see some of the more in-depth work on benthic habitat mapping and species distribution modeling from Jessica, Dave, and Ethan.
Great. So hopefully you can both see me and see the slides and presentation view again, Jamie?
Yes.
Okay, great. So we'll get started. So I'm going to start our Canadian Maritimes region deeper dive on fish habitat science. I'll start by giving a bit of a deeper dive on the benthic habitat mapping and benthoscapes, and then I'll pass it over to Dave Keith, who will cover the integration of benthic habitat maps into stock assessment and spatiotemporal species distribution models, and then Ethan Lawler from Dalhousie University will speak to species distribution modeling with the novel R package, which he developed called STARVE. So as mentioned in the overview presentation, for benthic habitat mapping, we focus on the benthoscape approach, which can be thought of as benthic landscapes, analogous to terrestrial landscapes, which represent the biophysical habitat classifications of the seafloor. So benthoscape mapping is conducted in areas where there's multi-beam data, and historically an area wasn't originally multi-beamed in order to derive a benthoscape map, but instead we would leverage multi-beam data from areas where it was already collected. However, with some new projects targeted for marine conservation, multi-beam data collection to feed the process of benthoscape mapping is part of the original project planning now for an area. And I seem to be stuck with my cursor. There we go. All right. So the areas that you see in red are areas where benthoscape maps have been derived, and areas in blue are ongoing benthoscape projects, with the Georges Bank benthoscape project slated to finish up in the spring of 2024. And the Eastern Shore Islands area is shown in the dashed line, since this is still in the kind of exploratory beginning phases for if benthoscapes will proceed for there. And I just want to mention that this work really wouldn't be possible with Dr. Craig Brown and his Seascape Ecology and Mapping Lab at Dalhousie University, with which we work very closely with. So, oops, I seem to have, there we go, yeah. All right, so in order to create a habitat map, two main data types are required. One being the continuous environmental data. So for terrestrial mapping, this is typically high resolution satellite imagery, but for the seafloor mapping, we are mostly limited to acoustic data, so bathymetry and backscatter and their derivatives. But we can also use, of course, continuous oceanographic data, so such as oceanographic model output. So here, for example, is shear velocity. And we also require ground truth data. So this allows us to verify the acoustic data and provide a fine scale insight into the community patterns that exist on the seafloor. And we have mainly depended on imagery, so camera or video. And as shown here, these are the locations within the Bay of Fundy that were ground truth with a high resolution 4K video from a drop camera. So subjective mapping, or by eye mapping, is an accepted method of mapping and is considered to be the conventional method of mapping, but interpretations and map outputs may vary depending on the expertise of map producers. And more recent developments in objective methods are becoming increasingly accurate and useful for producing fine-scale biophysical maps of the seafloor. And objective mapping provides a reproducible method to mapping and eliminates the subjectivity that can arise from different levels of expertise and knowledge base. And so in the Bay of Fundy, the benthoscape map was produced using object-based image analysis and fuzzy C-means clustering. So I'll just briefly walk through an example of how a benthoscape map is derived using the Bay of Fundy benthoscape as an example, and more details can be found in Wilson et al. 2021. So the multi-beam bathymetry and backscatter data were segmented using object-based image analysis to identify areas of homogeneous characteristics. And then other derived variables such as slope and shear stress were combined with the multi-beam data in a fuzzy clustering algorithm to group the segmented objects. Separately, each of the images extracted from the video data were then assigned a biophysical classification based on the dominant substrate type and the megafauna observed. And the classified imagery and acoustic data were then combined using an error matrix approach, with the final classification based on the most frequent and best matched biophysical classes. And this final classification was considered to be the benthoscape map. So once developed, these maps have many applications, and for us to determine the potential utility of the benthoscape map in relation to explaining patterns in sea scallop, which is our kind of primary day job focus, we overlaid the logbook positions. So these that you see in black are the logbook positions from the scallop fishery in the Bay of Fundy. And we can clearly see that there's very fine scale patterns in fishing activity as they correlate to different benthoscape categories. So looking more closely at the relationship between the fishery and the benthoscape classes, we see that the fishing effort is primarily in the mixed sediment, which is your brown, and gravelly sand, which is the green classes. So why is the fishery focusing on these areas? Well, the benthoscape habitat categories also explain the patterns that we see in the scallop survey data. So this is fishery independent data. And this helps in turn explain the spatial patterns that we're seeing in the fishery. So what we observe is the probability of a non-zero tow. So that's the probability that your tow has at least one or more scallop in it. This is lower in the sand, silt, and tidally scoured mixed sediment types, which you can see here on this plot to the right. So knowing this information, how does one explicitly harness the source of data, or the source of information to improve stock assessment? So I'm going to hand it over to Dave to discuss how we've done that.
Thanks, Jessica. I think that's the right.
Yep, I see it.
Do
You're
y'all
good.
see it? Good, good. So weird they make you lose your screen. Anyway, thanks. I was Dave Keith, research scientist in the scallop unit, work with Jessica on all this fun stuff. So what do we do with that information? So lots of things we can do. First thing we did is we looked at that delineation and thought about it in terms of where is the actual stock that we're interested in, that we care about, that the fishermen care about. And of course, because scallop's sedentary, that makes this easier than for other species for sure. But effectively what we did is we said, well, there's this large areas here. I hope you can see my laser pointer mouse thing, and up in this area. And so in these two areas, we went, well, there's not many scallop there, but it is a lot of area. It's not somewhere that they fish. And so what we did for our assessment model is, well, we'll remove these areas. I mean, the risk in a spatial model of keeping these areas is you have a low biomass that's not relevant to the fishery. It's essentially they're sink areas. But a low probability of seeing these things across a large area means you get a lot of biomass, and that's biomass we don't want to have in our assessment because it's not relative to the stock status itself. The other piece where we can bring this in is in catchability. So catchability is basically you're towing along the bottom, and it's the proportion of the things that the gear would have encountered that actually end up on the deck. So in this example, we caught 3 scallop out of 10. So the catchability here would be 0.3, whereas on this bottom type, we caught 6 out of 10. And so the catchability here would be 0.6 in our toy example. And so Miller 2019 had done some empirical experiments that gets us at estimates of what catchability is in different sediment types. And so we integrate that into our model structure as well. And what we came up with is what we call SHABAM, a spatially explicit habitat-based assessment model, where we both account for the catchability issues and the probability of encounter as well. And so how we do this is within a hierarchical framework. And so here we have multiple sources of variability included. I think most people hopefully are familiar with the idea of a hierarchical framework here. And essentially, so we have the observation level. So we have observed inputs from our survey tows, and we have error around that. That's integrated into the model. And then on the process side, the biomass, population dynamics and the error around that are integrated into our model framework. I'll talk mostly about the observation side here. So the traditional observation equation, I'll show it in a sec, but effectively we're taking survey biomass, so the biomass you see from your survey, and you're trying to get that into a population level biomass estimate because that's what you're interested in. And so how you do that is this catchability term. So you take your survey index, and then you take your population level biomass, and there's a proportionality constant there. And that Q is catchability. There's also uncertainty around this, as mentioned earlier. And so that's how you traditionally do it in math. This is how Raphael McDonald, who asked the question a second ago, managed to pull it off for us in maths. And effectively, our catchability now becomes habitat and spatial informed. So it's informed by habitat and where it is in space. And that probability of non-zero tow is also informed by the same information. So yeah, so both these things now account for your sediment type, the bentoscape map that we have there. And what comes out on the other side, oh, sorry, yeah, one more. And so what you'd see and visually, a traditional queue is basically there's one queue for the whole area that you're going to have your assessment done at. You get one number for this whole area. What we get with this method is actually spatially habitat-based informed queues. And so we have queues for a whole subset of the area. And this all is naturally coming out from the Miller information as our priors, and then being refined within the model itself based on the data that we have. And so what we've found is this really does an excellent job of capturing spatial patterns. I'm just showing biomass here, but it also, we have spatial patterns in recruitment and natural mortality. So and we have the spatial patterns in fishery removals too. So the really key components for our model structure in terms of productivity. And what's interesting in the Bay of Fundy Scallop example we have is, you see in 2002, there was this really small area. So this is biomass density, but a very small area of very, very high biomass. And the rest of the area had lower biomass relative to this. We move forward, this was 2002. In 2018, we see, well, biomass is actually quite spread out compared to 2002. Yeah, it's quite spread out across the domain. And so how did the two models handle this? Well, we get a total biomass estimate here. And in 2002, our traditional non-spatial modeling framework said basically that's the highest biomass, 2003, but effectively that's the highest biomass we ever saw was here. Whereas our spatial model goes, well, that was really concentrated in a small area and there was a lot of lower biomass areas. So I don't think it was quite that high. And we pop forward to 2018 and you see actually the pattern reverses. So when it's evenly spread across the domain, the habitat informed spatial model gives us a higher biomass estimate than a standard traditional model is using a stratified estimator. And so that's an interesting change in our perception of the population dynamics. We come back to this and go, well, actually, we're probably in the most productive period ever in recent years, whereas before we thought 2002 was just an amazing period. So that's one area that we get some interest. That's the main thing we get out of those. Well, sorry, Raf, not the main thing, but that's an important component that we get out of these models. So I'm going to switch gears here and talk about species distribution modeling. And so this is work that Nathan Hebert has done for us. And so I'm going to summarize kind of the three things we found from this, and Jessica hinted at some of these already. First, the results depend on whether you account for spatial dependence or not. Spatial dependence is very, very important in these models. And historically, certainly most species distribution models did not account for spatial dependence, and it really changes your perception of the world when you account for it. The spatial temporal covariates, I think Raquel put a message in there saying they're seeing the same thing in Newfoundland. We've seen this in Nathan's work. I've seen this in some other work that I've done. The spatial temporal component is really explaining most sometimes of the variability compared to the environmental covariates alone. So the space defines how the stock varies more or less. And to me, that's probably unknown environmental relationships going on, right? So it's some environmental forcings that we don't fully understand. And then the really interesting piece that came out of this was that, well, one of many, but a really interesting piece is the significant environmental variables actually depend on the model framework that you're using. So you get different significant environmental covariates or variables coming out of your model, depending whether you're using a GAM, whether you're using a spatial temporal GAM, or whether you're using a different spatial method. And so our advice on that right now is you really should be looking at multiple models to be sure your environmental variable is significant across models. And so the case study I'll show you that kind of hammers home these 3 points is a species distribution modeling that we've done for orange-footed sea cucumber on a Scotian shelf. We fit equivalent models to the same environmental terms with smooths using 4 approaches, and they're in a delta model as well. So we had a non-spatial GAM, we had a GAM with spatial temporal smooth functions, We had a spatial temporal generalized linear mixed model using nearest neighbor Gaussian processes. Ethan's going to talk about this in about 3 slides when I finish. This is the STARV package, very fast fitting and very interesting methodology to get spatial temporal information into these models. And then slightly older, I think, than what Ethan's developed at least, is this generalized linear mixed model framework using stochastic partial differential equations, Gaussian Markov random fields. This is the SDM-TMB package that Sean Anderson in the Pacific region for DFO has built, and very slick and very quick as well. So accounting for spatial dependence. So these are, I'm going to start with the 3 spatial models. We have a spatial GAM, we have Ethan's STARV model, and we have a Gaussian Markov random field, the GERMF model, as I will call it. And what we see in these 3 models is really the patterns are quite similar. We don't see a lot of difference. Either of the 3 methods really drives home a very similar picture if this is if the picture that you're looking at is trying to understand what the distribution of this species is. Again, this is sea cucumber example. The environmental variables in this example are all the same for each model because we want to compare apples to apples. But the real outlier here is the non-spatial GAM. And that non-spatial GAM we see especially along edges in areas where this model would predict very low CPUE, which is effectively the catch per unit effort here, right, from a survey. These models predict it's actually much higher. So your estimate here, if you were turning this into a biomass estimate, would be much higher, I should say, for non-spatial GAMs than for any of our spatial GAMs. And the other thing, if you're talking to a fisherman and saying, well, actually, there's going to be a lot of sea cucumber right here, if I use this based on the environmental covariates, well, in fact, they aren't there. And so when we look at all the spatial methods, there isn't actually there. And you see this whole way down this area where there aren't actually many sea cucumbers, but the non-spatial GAM is predicting a relatively high biomass. And so this really interesting and I think important difference between our traditional non-spatial methods. And I'd be really interested to check this out with something, different species, different distribution frameworks that don't have space in them with these kind of spatial species distribution models to see how different they are. But that sent some warning bells off. The other thing that really, I think, has a big impact down the road to think about. So we have our 4 models here. I'm colorblind, so this color, where my mouse is non-spatial GAM. I think this is blue. This is our spatial GAM. It's definitely black. That's our STARV model. And that color, which may be red or green, is our Gaussian Markov random field model. So our 3 spatial models really generally for the environmental covariates give the same response. Right? The very similar response in all 3, except this one, and I think it's in STARV, there's a little bit different response for the bottom temperature range environmental covariate. But our non-spatial GAM, so when we remove space, the patterns are often reversed. So for example, here for our depth covariate, this one, I mean, the directionality is fairly similar, but this would say my likelihood of seeing or the biomass declines really rapidly at the start, where this other one says actually it doesn't decline that much at the start, it really declines towards the end. And so you get these opposite patterns, you have different directionality here for the bottom stress between the spatial models and the non-spatial models. And in this case, I would say this term, I think, and Nathan can correct me if this term from the non-spatial model actually is non-significant. So this wouldn't even be in there. So you wouldn't say bottom stress is a significant predictor in a non-spatial GAM, whereas for all the spatial models, actually it is, and it has a negative impact on your biomass estimates. So it's, yeah, it's very interesting that we're seeing these differences, and I think they're probably rather general. And if you just think about using a non-spatial GAM to do your species distribution models, I'm going to tell you it's a different suite of environmental predictors than if I use a spatial model. And when trying to predict the future, that seems pretty important to me. So I'll wrap with that. I think that's all I'm supposed to say, and throw it over to Ethan. You can steal the screen.
Will do, thank you. So while I'm pulling it up, my name is Ethan Lawler. I'm a postdoctoral researcher at Dalhousie University. And I work pretty closely with everybody over at DFO. So I'll be talking just basically a lightning cell on the STARV package. So species distribution modeling with this package, it's in R. You can go get it on GitHub. And the acronym STARV comes from Spatiotemporal Analysis of Research Survey Data. The analysis goals for using this package are if you want to create maps of predictions or forecasts, like Dave was just talking about, and performing covariate inference. And there were three kind of guiding design principles for this package. So the first one is I wanted to use a simple user interface. So I wanted this package to be usable and accessible for many people to use. I wanted to use native use of spatial data formats. And so if you have, if you're used to working in ArcGIS or other GIS software, exporting your data from that into R and then using the STARV package is a pretty seamless experience. And then also computational efficiency is a huge concern when you're doing spatial modeling and spatial temporal modeling. And so getting these models to run quickly so that you can focus more on the actual modeling instead of just waiting for things to run. So the applicable data sources for the package. The spatial information consists of latitude and longitude points. They can be projected or unprojected, either is fine. And these would be either a fixed station design or random sampling design. And random sampling that would include stratified random sampling, like is common in surveys. What it can't do is it can't account for preferential sampling. So if you think of like industry data where they're fishing where they think the fish are, currently the package cannot handle that. As far as the time information, you're looking at discrete time, so daily, monthly, or yearly, something like that. And then it can handle a range of response variables. So it can be count data, continuous data, for example, biomass, be presence or absence. It can be binomial. So I've really tried to make it flexible for a number of different types of data. So just giving a brief example of what this package can do, I have a haddock survey. So this was just over 3,500 observations of tow weight across 20 years. I think it was 1997 to 2017, something like that. So we took this tow weight, its biomass, but since it's fisheries data, we also have a large number of zeros. And so to account for those zeros, we used a Tweedy distribution. We also used a barrier to account for the Nova Scotia landmass because we have some observations up in the Bay of Fundy all the way across the Scotian Shelf. So we needed to account for the fact that fish can't cross land. We also want to make our predictions. So the predictions for this example, we made for a 50 row by 50 column raster. And we did those raster predictions for each of the 20 years in the data set. And then for the computational efficiency, the timing for these models to fit it was just under 3 minutes. And then predictions was just over 3 minutes. So you're looking at about 6 minutes total for this entire analysis. So this is what the data looks like. This is just a pretty standard survey. So you can see where all of the points are for this survey. And we want to take this data, this total weight, and make our predictions. And to do that, we have these. And you can do a number of things with these maps of predictions. I mean, the first thing you can do is just easier to see what the trends are that are coming from your data. If you have environmental covariates and you're not sure which you want to include in the model, it's easier to compare these maps to maps of your covariates to test some hypotheses. And then you can also aggregate these. So if you want like a single number, if you want to do like a total biomass across the shelf or an index of that, you can aggregate these across space, or you can identify hotspot zones. So if you take like the top 10% of the predictions, you can do a number of useful things with these. How to use this package? I'll just go really briefly. I'll compare my package STARV to kind of the most popular competitor, which would be our INLA, which if you've used spatial methods before, you've probably at least heard of. So this comparison was made using a Carolina Wren survey. So this was just birds in the state of Missouri. And this had 783 observations of wren counts across 21 years. So to analyze this data, including fitting the model and getting your predictions, with the STAR package took 15 lines of code. So the first line is just loading the package. Line 3 through 8, that's fitting the model. And I tried my best to make it familiar to people who have used packages such as LM or GLM. And so, for example, on line 4, you have your model formula. Line 5 is the data that you want to analyze. And line 6 is your response distribution. Line 10 is just creating a template raster to tell the package where you want to predict. And then lines 12 through 15 is making the predictions on that template raster using the fitted model. So comparing my code, 15 lines, to our inlaw, I actually needed an entire scroll bar to fit this analysis in the package or in the presentation. I'm not going to go over everything. I'll just note that if I scroll all the way down, it takes 101 lines of code to do the exact same analysis. So the things I'll say is that you need to really learn inlaw to make sure you're doing it right. And then anytime you're getting into large lines of code, you're going to run into bugs, and it's just going to take more time to code as well. And then comparing the computational efficiency, the first 2 rows are the fitting time, prediction time, and memory usage for inlaw. I think the default for inlaw is to use parallel. So for example, fitting a model took 20 seconds. And then the last 3 lines are the STAR package. And this k equals 30, k equals 10, k equals 3, that's a tuning parameter. The default is 10, and that took 5 seconds. And so in addition to saving time and being more sure that you're coding the models correctly, you're also taking about 25% of the time to actually fit the model. So for more information, there's my GitHub page. So Lawler EM/starve. The paper is published in Methods in Ecology and Evolution that came out earlier this year. And then I just wanted to mention some work in progress or companion projects. So I'm working on spatiotemporal growth curves, and there's going to be another package for that that's going to work very similarly to the STAR package. And then I'm also working on spatiotemporal stock assessment models. And both of these will be habitat informed. So habitat is going to be a huge part of all of these. So that is my part of the presentation. So I'm going to stop sharing, and I'm sure when we have questions, I'm happy to answer all of them.
Thanks, Ethan. Okay, so I think that concludes the presentation portion of the fish habitat seminar. So thank you to all the speakers. So Laurel, Tori, Chris, Jessica, Dave, Ethan. And with that, I'm going to turn the recording off.